The manufacturing of Prevnar, a highly complex and technologically advanced vaccine, was made possible when precise methods were devised to consistently and accurately produce the 7 individual vaccines for the 7 most common serotypes of pneumococcus, and to combine them into a single dose using a stable, reproducible, and functional process. The process of conjugating 7 serotypes is considerably more complex than conjugating 1 serotype, as is done with other licensed conjugate vaccines. Each serotype must be conjugated and formulated in precise quantities to achieve the clinical immunologic response to all serotypes in the vaccine. The manufacturing process for Prevnar begins with the production of the protein carrier CRM197, which is a non-toxic variant of a diphtheria bacterial protein. These bacteria are grown in large fermenters at a facility of Wyeth. The bacteria transport the CRM197 protein into the surrounding liquid growth medium. Once they have reached the desired level of growth, the bacteria are separated from the growth medium, which contains the protein carrier. The CRM197 protein is then separated from the much larger bacteria using ultrafiltration. In this process, the bacterial cell debris remains on the filter and the liquid containing the CRM197 protein passes through. The proteins contained within the liquid are concentrated and washed to remove debris. The CRM197 protein is purified further using a highly efficient process called chromatography. The purified protein is then frozen and stored, awaiting conjugation to the saccharides derived from the pneumococcal bacteria. The saccharides for this conjugation process are isolated at a separate Wyeth manufacturing facility. The 7 different serotypes of pneumococcus in Prevnar are independently grown in large fermenters. After having reached the desired level of growth, the pneumococcal bacteria are treated with detergents to release all of the bacterial components into the liquid growth medium. The saccharides contained within the liquid are then purified to remove contaminating bacterial components, first by filtration and then by separation on chromatography columns. The purification process takes more than 7 days for each saccharide, but the ultimate result is a purified saccharide derived from each of the 7 vaccine serotypes of pneumococcus. The manufacturing facility produces only 1 serotype of saccharide at a time. Therefore, this process must be repeated 7 times to produce 1 batch of Prevnar. The next step of this complex manufacturing process occurs when the saccharides and the carrier protein are brought together for conjugation. Each of the 7 purified saccharides is chemically activated and subsequently filtered to clear unnecessary small molecules. The activated saccharides are then independently linked to the purified CRM197 protein under specific conditions that vary significantly according to serotype. During this process, the saccharides conjugate or attach to the CRM197 carrier protein. Each conjugation process takes up to a week to complete. In order to produce Prevnar, 7 separate conjugations must be performed. Next, each of the 7 separate conjugates that represent the 7 serotypes found in Prevnar is combined to create the vaccine in bulk liquid form. Extensive quality testing is performed on this batch of conjugated material. These tests are designed to help ensure that Prevnar meets the requirements for purity, potency, and safety. Overall, more than 300 separate quality control tests are conducted during the production of each batch of vaccine. After passing extensive quality control tests on a manufacturing line, Prevnar is dispensed into vials in preparation for final packing, labeling, and distribution. Additional quality tests are conducted on the finished product. Throughout the supply chain process, Prevnar is maintained in a cold chain environment that monitors the temperature of the product to help ensure its integrity. The complete production process cycle time for each batch of Prevnar spans 9 to 12 months, involves multiple manufacturing facilities, and employs hundreds of trained professionals. There are risks associated with all vaccines, including Hib titer. Hib titer will not protect against H. influenza other than type B strains, nor will it protect against other microorganisms which cause meningitis or septic disease. Hypersensitivity to any component of the vaccine, including diphtheria toxoid, is a contraindication to its use. Please see prescribing information for indications and usage, dosage and administration, and safety information. Risks are associated with all vaccines, including Prevnar. Hypersensitivity to any vaccine component is a contraindication to its use. Prevnar does not provide 100% protection against vaccine serotypes or protect against non-vaccine serotypes. See prescribing information for indications and usage, dosage and administration, and safety information.